Well, welcome back everybody to Coronavirus Shutdown Part 2. I hope that you guys have been having a nice day and you're not too bored at home. This is a little early. You'll get to see this tonight at 7. So I'm going to go through the guided practice for today. So if I'm going to look at question number 1, it says, what is the essential nature of the Davison-Denali model of the membrane structure? So when you're looking at a membrane, we do know that it's going to be comprised primarily of 2 parts, and those 2 parts are going to be proteins and lipids. Now, the Davison-Denali model, do not have corona. They knew that these proteins and lipids made up the model, but they weren't sure exactly how. They knew that the proteins had a function and it had to be on the outside and inside of the membrane, but they weren't sure exactly how it was comprised, or at least in how it was arranged. We now know that their model was inaccurate, but what can you do? This was in the infancy of science, or at least in terms of modern science, and being able to investigate these structures. So their proposal was that you had in the interior, you had lipids. That would be the fluid potentially part of the membrane, and on the outside you had proteins. And so what they thought was it was kind of like a sandwich, that you'd have proteins like bread on the outside, and then you would have the lipids on the inside of this. And so this would be what the arrangement should look like. Proteins here, and then lipids, phosphate head, fatty acid tails, and they were on the interior. So let's take a look at our answer choices. Is a phospholipid monolayer? Well, they didn't think it was a monolayer, so they knew it was a bilayer, at least 2 layers of lipids, just because of the properties of lipids being hydrophobic and hydrophilic. Hydrophobic tails and hydrophilic heads. Hydrophilic meaning loving, phobic meaning fearing. Let's look at B. A layer of protein sandwiched between 2 layers of lipids. That's not what they proposed. They proposed that the proteins are on the outside, so B is incorrect. A phospholipid bilayer with intrinsic and extrinsic proteins. Sounds pretty good because we got intrinsic and extrinsic, but I'm not sure that that's going to actually be it. Let's look at D. A layer of lipids sandwiched between 2 layers of proteins. Yes, that would be true. That would absolutely be true. So I'm going to go with D, and I'm going to cancel out A, B, and C. Let's look at number 2. Number 2 says describe the functions of proteins in the cell membrane. So I taught you guys the acronym TRACY, so I'm going to go ahead and put that down. And it says describe, so obviously you're going to have to use more than 1 sentence to do this, but I'm just going to illustrate what this acronym stands for. So first was going to be for transport. Transport, this would be like protein channels and such. Protein channels. So things can flow through. So this is mostly going to be for diffusion. And then recognition, you're going to have cells that are going to be able to recognize one another. So you're going to have recognition. Cells would know that you are self or non-self. So in this case, like a virus were to get into you, you would be able to recognize based on its antigen. So this would be antigens or receptors. Okay, and receptor speaking, let's just go here to communication. So communication would be another way these proteins work. So cells have to be able to communicate, and they use receptors to do this. Incidentally, the ACE2 receptor and your nematocyte number 2 is how COVID-19 actually attaches with its antigen. And this would be adhesion. Your cells need to be able to stick, be able to come in contact with one to another and adhere to one another. And then you have intracellular joinings, which is coincides with this. So remember your intercellular and extracellular matrix. In that case, I'm going to kind of draw two cells here. Nucleus. From here, you have the cytoskeleton, and they're able to connect to one another. They'll be anchored to proteins embedded here, and they allow for those joinings. So here are your joinings between cells. So those are my intercellular joinings. And then finally, we get enzymatic function. And enzymatic functions are going to allow for reactions to take place. This could also be like proton pumps, and then also allowing for reactions. So reactions can occur. So I'm trying to think of, like, if you're going to start a transductive pathway, you would have an enzymatic functioning type protein. You would have a ligand that binds to this, and it causes a cascade of these signals that goes to the nucleus and then allows you to express some certain gene. But this ligand would bind to this protein that has a form of enzymatic function. Okay. Number 3, let's see, explain how hydrophobic and hydrophilic properties contribute to the arrangement of molecules in a membrane. So let's just break this down into parts. So let's think about sides. So I'm going to start with sides. So we know that we have a hydrophobic side, and we have a hydrophilic side. I'd put that as my point number 1 and start there. And let's break this down and talk about the parts. So parts that are hydrophilic and hydrophobic. So I know my hydrophobic, let's do that first, hydrophobic side, that is going to be, on the hydrophobic side, that's going to be my fatty acids. They are scared of water. They do not like the water. Hydrophilic side, hydrophilic side, this is going to be your phosphate heads. They like the water. They're going to face the interstitial fluid on the outside of the cell as well as the cytoplasm in. So let's draw that really quick. And this is going to be the outside of the cell. This would be my interstitial fluid. And this inside is going to be the cytoplasm. Now both of these are going to be aqueous solutions. So these are going to be an aqueous solution. Not perfect. This is also going to be aqueous solution, meaning it's water-based solution. And these fatty acids here, they're going to face the inside away from the water, and the phosphate heads are going to face towards the water, and these are away from the water or the cytoplasm or interstitial fluid. So let's think about this in terms of behavior. It says it's worth 7 marks, so there's a lot of things that we really need to think about talking about in this way. I would say the same thing. You're talking about the parts here, that they have fatty acids and phosphates, and then what I just explained for behavior and how that they work. Because of this, they make a lipid bilayer. Bilayer meaning that it has 2 sides. You have fatty acids that are going to be on the outside of the membrane, fatty acids that are going to be on the inside of the membrane, at least towards the cell, and this would be outside of the cell. So this would be like blood or mucus or something along those lines, and kind of fluid surrounding the cell, and this is going to be the interior of the cell. So you could think of them in terms of this way as well. You might, again, direction, you could come back, you're talking about 7 marks, so we really got to capitalize on this. Talk about the direction. So the direction that I just talked about. So in this case, the phosphates on the outside of the membrane, and the fatty acids inside the membrane. Let's see, what else? Cholesterol, we didn't talk about cholesterol. Cholesterol could have an effect. It says hydrophobic and hydrophilic properties. I throw cholesterol in there. So cholesterol, let's bring that down here so it doesn't confuse it with direction. Cholesterol is going to be an animal. Most animal, not or all animal cells and some other organisms have cholesterol in their membrane. So animal cells, and this allows for a lower freezing point. So we know that lipids, these lipids are like fats. Fats, when they get a low enough temperature, clump and become a solid. Cholesterol, not as much. And so it raises that and lowers that temperature at which that they'll crystallize or they'll freeze. And that allows you to survive in lower temperature, especially for organisms that would be exothermic. It also helps with diffusion too. Yeah, it prevents some particles from diffusing into or out of the cell. So diffusing. What else could I say? So we could address proteins. So proteins are comprised. Their monomer is going to be the monomers are amino acids. And being that they're amino acids, some amino acids are going to be hydrophobic and some are going to be hydrophilic. And so that's going to allow that membrane to have different parts of the protein in different areas. So in this case, this part of the protein, you would know that this is going to be a hydrophilic part. This is going to be let's just go here and say hydrophilic. And this as well is also going to be hydrophilic. But this region here, sandwiched between these two parts, that's going to be hydrophobic. It would have to be because it's on the interior of the membrane, away from the interstellar fluid here or the cytoplasm here, and dissolved inside of the membrane where my fatty acids are, which are also going to be hydrophobic. Okie doke. So can't really think of much else to say. That's 1, 2, 3, 4, 5, 6. There's got to be a 7th mark in there. What else could we say about this? Well, you could also say in terms of your proteins that this will be an integral protein. It goes the entire distance, so this would be an integral protein. And it's going to have hydrophobic and philic portions. But you're also going to have peripheral proteins. And these peripheral proteins would be mostly, they're going to be, let's say that's an integral protein here, and it's facing only on one side of the membrane. So this being the outside, this being the inside, and it's only situated on the outside. So these are going to be mostly comprised of hydrophilic amino acids. Okay, that's an extra mark. 1, 2, 3, 4, 5, 6, 7. I think I got all 7 marks. Let's go to Oh, another one of our calculations. And this time I think I'll make it a little, hopefully I'll make it easier. Let me look at the centering of this. Yeah, you'll be able to see it. So last time I didn't have this where you guys could see my calculation. So magnification is equal to the image size divided by the actual size. So this is the image size divided by the actual size. So let's take a look at our question. See this here. Let's see. So we got to calculate the magnification. So what is the magnification? We do know that the actual size is 25 micrometers. So I need to get a ruler. And I'm going on the inside of these bars, so from this corner to this corner, and that looks to be about 2.2 centimeters. But I need to convert to get to nanometers. So 2.2 centimeters is equal to 22 millimeters, and you go up by a factor of 1,000, so it'll be 22,000 micrometers. Shoot, I don't have a calculator, it's on my telephone. That sucks. Again, I didn't make the same mistake. Excuse me. Sorry about the pause. Let's see, 22,000 divided by 25, 800 equals 880. So this would be my magnification. So we are 880 times, so almost 900. I'm sure they'll give you a bit of a range. Oh, they're crazy. Okay, so let's take a look at number 4C. Explain how the amphiphilic nature or amphipathic nature of phospholipids allows them to form bilayers. But just essentially went through this. So remember, amphi means both. So both. In this case, they're talking about hydrophobic and hydrophilic. So I would say phosphate heads are hydrophilic, fatty acid tails, hydrophobic, and then therefore creates, and I'm abbreviating this, creates a lipid bilayer, or two layers. Two layers. Okay, guys, that is for today. I will have another one for you guys on Tuesday. I hope you're enjoying your break. Don't forget, you can contact me, and I may see if I can set up an account and send you guys an email where you guys can Skype in and ask me more questions about this. Maybe Skype, or there's another program that Mr. Leonard and I are going to take a look at tomorrow, a way that I can meet with you guys online as a group, that we can go over some of these questions as well. I'll talk to you guys after a while. I miss you. Bye.